Okay, let's talk about two different biotechnological techniques, recombinant DNA and bacterial transformation. Recombinant DNA is when a scientist has DNA and they decide that they want a particular gene or gene of interest from that DNA. So let's say right here is a gene of interest that they want to replicate and grow. Let's say that it codes for insulin, right? So they want to cut this gene out. How would they do that? In a previous video, we talked about something called restriction enzymes. Now, restriction enzymes go along the DNA and they look for particular patterns of those nitrogenous base pairs. They're going to look for a pattern. And once they find that pattern, so let's pretend that we have our DNA here. So I actually want to draw my DNA in blue. So here's my DNA and our base pairs. And we have our restriction that comes along. It finds a particular code. And there are many different types of restriction enzymes. It finds a code and it cuts through those hydrogen bonds and comes out the other side. And then it does that again. So let's make this DNA just a little bit longer. So it does that again. Okay. And it leaves these things called sticky ends. If we were to draw that out, actually would look something like that. Now, once you have your gene of interest, you've cut it out using the same restriction enzyme. So that's really important. The same restriction enzyme is being used to cut on both ends. Why is that really important? When you go to put it into a vessel that you want to grow it, it needs to have the same pattern. It needs to match up. So what that's going to be grown in is a plasmid. Here, so we have our plasmid, and a plasmid is just a circular piece of DNA. Most of the time it's found in bacteria. So here's our plasmid. Okay. And so the restriction enzyme that was used to cut your cellular DNA is now going to be used to cut your plasmid. So it's going to cut just like this. And it's going to leave those same sticky ends, which is going to allow you to put this cellular DNA inside of your plasmids. This is now going to look something like this. Right? So this is your new genetically engineered plasmid with the gene of interest. Now, sometimes scientists will also genetically engineer their plasmids to contain genes that are antibiotic resistant. Now, antibiotics are what kill bacteria, and normally they're grown on a petri dish in bacteria. But they want to make sure that the genes that they're growing, the plasmid, only grow in the environment that they designed. And what does that mean? So let's kind of talk about that a little bit. This is where we have bacterial transformation. What we're going to do, we have our new plasmid, and we're going to take a vial. And in our vial, we have a couple of things. We are going to add transformation solution, which is just calcium chloride. Transformation solution. We're also going to add our E. coli. Now, E. coli is going to be our organism that our new plasmid is going to be put into. All right, so we have transformation solution, we have E. coli. Okay, so this is just calcium chloride. And then we need the plasmid, right? So let's just kind of add the plasmid in there. Okay, so we have our plasmid. Then what we're going to do is something called heat shock. We're going to have this on ice. So it's going to be ice and then heat very quickly, and then ice again. What's that going to do? The heat is going to allow pores. So that's going to allow the bacteria. So let's just say this is your bacteria, this is your prokaryote, or maybe, yeah, we'll say that it has a little flagella. And so the cell wall actually will have these become porous. And the transformation solution, so again, this is calcium chloride, that neutralizes the DNA. Calcium chloride has a positive charge. And as we know, bacteria is negative. So that's going to allow this new ring of plasmid to slip into the bacteria during the heat shock. And then the ice, it's going to close up those pores, and it's going to allow the plasmid to be inside our bacteria. And then what are we going to do? So we've ice, heat, ice, and then we've got to give it some luria broth. Luria broth is like chicken soup for bacteria. It makes them grow and nice and happy. So then you've got LB, which is luria broth. And then you're going to put it on a petri dish, and we're going to grow it. And from there, you are going to be able to then isolate out that particular gene. Normally, these are grown on an antibiotic plate. So let's just say this has ampicillin, which is a type of antibiotic, so that it only grows our new bacteria with that specialized plasmid. Any bacteria that didn't take in that plasmid would die. So we're targeting that specific gene.